Hi, welcome you all back to another video in Microbiological Insights. So in this video we will be going to discuss about the most important topic in microbial structure, that is about the plasma membrane or cytoplasmic membrane. These are the subheadings we are going to discuss here, that is about the definition of plasma membrane and about the fluid mosaic model structure of plasma membrane and the functions of plasma membrane. So if you want to watch this video in Tamil, you can please subscribe this microbiological concepts channel. Okay, now let's move into the video. What is plasma membrane? The plasma membrane is a thin structure. Where it is located? It lies inside the cell wall and it encloses the cytoplasmic constituents. So this layer is made up of mainly phospholipids and some protein molecules are embedded inside this phospholipids. So it's like this structure, no? These are all phospholipids and these are the proteins which is inserted into this membrane. So we can see this type of structure in all the living cells. When we see the prokaryotic plasma membrane means the thickness is about 75 nanometer and the prokaryotic plasma membrane consists of primarily about 20 to 30% of phospholipids and about 60 to 70% of proteins. When we see the eukaryotic plasma membrane, in addition to the phospholipids and proteins, this eukaryotic plasma membrane also contains carbohydrates and sterols. The example of sterol is cholesterol. So these two molecules are present in addition to the phospholipids and proteins. So when we compare both the prokaryotic membrane and eukaryotic membrane means the prokaryotic membrane is less rigid because it doesn't contain sterols, but the eukaryotic membrane is highly rigid in nature because these eukaryotic membrane have sterols. And there is another one exception, prokaryotic mycoplasma. You know about this mycoplasma, no? This mycoplasma lacks the cell wall. It has the plasma membrane. In the plasma membrane of mycoplasma, membrane sterols are present. Next, we move on to the fluid mosaic model structure of plasma membrane. This is the widely accepted model for the plasma membrane structure. And this structure was proposed by Singer and Nicholson in the year 1972. They proposed that this membrane are made up of lipid bilayer within which the protein molecules are embedded, which looks like a fluid mosaic structure. The electron microscopic studies showed that the prokaryotic and eukaryotic membranes are made up of two layered structures. You can see the two dark thick lines here, and between these two lines you can find one white space, but it is not clearly seen here. When we see the structure here means you can see the continuous dark line with the white space in between the dark lines. This characteristic appearance showed us the membrane lipid is organized in two sheets of molecules which are arranged end to end. When we see the picture, this is the lipid bilayer, this is the peptidoglycan layer, and this is the outer membrane. The phospholipid molecules only arranged and it forms the lipid bilayer, no? Here you can see one layer of phospholipid molecule and this is the another layer of phospholipid molecule, which this both together form the lipid bilayer. So these two layers are parallel to each other, and this is the polar head and this is the non-polar fatty acid tails. When we take the each phospholipid molecule means you can see a polar head and a non-polar tail. This is polar head and this is non-polar tails. So this is the clear picture of one phospholipid molecule. This is one molecule and this is one molecule. You can see clearly the polar head here and this is non-polar tails. The polar head is hydrophilic in nature. Hydrophilic means it loves the water molecules. So it has an affinity to the water molecules. And these polar heads are made up of phosphate group and glycerol. And this non-polar group is hydrophobic in nature, and these are made up of mainly fatty acids. So here the polar heads are made up of phosphate group and glycerol. Here the non-polar tails are made up of fatty acids. Polar heads are soluble in water and the non-polar heads, because it is made up of lipids, no? So this is insoluble in nature. And this is the clear structure of one phospholipid molecule. When we see the polar heads and non-polar tails in the lipid bilayer, the polar heads are present on the two surfaces of the lipid bilayer, here and here. And the non-polar tails are present inside the bilayer. Here you can see the non-polar tails. And this is the clear structure of the plasma membrane lipid bilayer. And next we move on to the embedded proteins. The proteins which is embedded in the plasma membrane also have several functions. So sometimes this protein serves as channels for the transportation or movement of molecules from outside cell to inside cell or from inside to outside. Sometimes it acts as the receptor site for binding hormones and neurotransmitters, and sometimes it acts as structural proteins. These are the functions of embedded proteins. How these proteins are arranged? So there are some proteins termed as peripheral proteins. Peripheral means it can present in the surface. So it can present in the outer surface or sometimes it is attached to the inner surface. But it is not deeply inserted into the membrane. Just it present outside or inside the surface of the membrane. And this protein is soluble in aqueous solution. Aqueous solution means you know about that, no? The solution in which the solvent is water, that is aqueous solution. So this peripheral protein is soluble in aqueous solution, and this peripheral protein make about 20 to 30% of total membrane protein. And this peripheral protein also have many functions. Sometimes this peripheral protein acts as enzymes to catalyze a particular reaction, or sometimes it acts as a support or scaffold, and sometimes it acts as a mediator. And next we move on to the integral proteins. Already we discussed clearly about the peripheral proteins, and now these are the integral proteins. It constitutes 70 to 80% of total membrane proteins. And this protein fits deeply inside the membrane. So it cannot be easily removed from the membrane by mild treatment. No? If the lipid bilayer undergoes any disruption or any breakage means at that time only this integral proteins released from the plasma membrane. Otherwise it is not easy to remove it. Sometimes these integral proteins penetrate into the membrane completely. Completely it can present inside the membrane. In this case it is termed as transmembrane proteins. When we see the integral proteins, it sometimes acts as channels. Channels means it gives a way. It has some pores. So through that holes only some substances enter inside the cell or it can exit outside the cell. So it acts as channels. And sometimes the carbohydrate molecule is attached to many of the proteins and some of the lipids. So if the proteins attach to carbohydrates, we can call it as glycoproteins. And if the carbohydrates attach to lipids means we can call it as glycolipids. What's the role of these glycoproteins and glycolipids? These molecules helps to protect and lubricate the cell. What is lubrication? It can helps to move smoothly. No, that is lubrication. It also helps for the cell to cell interactions. So these are the functions of glycoproteins and glycolipids. And this is the clear structure of fluid mosaic model of plasma membrane. You can see clearly the peripheral protein here. It is attached sometimes in the inner surface, sometimes attached here also. So it may be present in the surface. And these are all integral proteins. Sometimes it can fix deeply inside the cell. And you can see a twisted structure here, no? This is hydrophobic alpha helix. And here one red molecule is seen. What is it? Hopanoid. This is nothing but a bacterial membrane lipids. Sometimes this hopanoid acts as sterol. And this hopanoid is not present in all the bacteria, no? It is present in methanotrophic bacteria, heterotrophic bacteria and cyanobacteria. But it is not present in all the bacterial groups. You can see the glycolipid here. Glycolipid means if a carbohydrate molecule comes and attached to the lipid molecule means we can call it as glycolipid. Here one oligosaccharide is here, no? So it is also a carbohydrate. So if the carbohydrate molecule comes and attached to the surface of a protein means we can call it as glycoproteins. So this is a glycoprotein. This is glycolipid. And when we see the structure, the phospholipids and protein molecules are not seen in one place. It is not static or stagnant in one place. It can move freely inside the membrane. So it can, sometimes it is present here, the protein is present here, sometimes it moves to this place. So it can move from one place to another inside the plasma membrane. Why these phospholipids and proteins move from one place to another inside the membrane? So to carry out the function only it can move. And when we see the structure of this phospholipid and lipid molecule, the phospholipids is here, no, arranged here, and these are the fatty acid tails. These are polar heads. So the fatty acid tails cling together. So the phospholipids in the presence of water, it can form a self-sealing layer. So if there is any repair or break or any cut occurs in the plasma membrane means this self-sealing bilayer repairs itself. So this is an important property of this structure, no? And when we see the structure, it is the plasma membrane is viscous in nature. Viscous means it is sticky, like olive oil. So because of this sticky nature only, this phospholipid and protein molecules can move. It is easy for them to carry out the special functions. So this dynamic arrangement or changing arrangement of both the phospholipids and proteins is termed as the fluid mosaic model structure. Okay, I think you clearly understand about this. Let us move on to the functions of plasma membrane. The first important function is selective permeability. The other name of selective permeability is semi-permeability. So from the name itself you can clearly understand that it can select particular molecules to pass through it. It can prevent some molecules from entering inside the membrane. No, this is selective permeability. And the permeability of the membrane depends on several factors. What are the factors? First, large molecules. The example of large molecules are proteins. The large molecules cannot easily pass through the channels of integral proteins because the channel size is small when compared to the protein size. So it cannot pass through the membrane. And when we take the smaller molecules such as water, oxygen, carbon dioxide and sugars, it can pass easily because the size is also small, which is equal to the size of the integral proteins channel size, no? So it can pass through it easily. But the ions enter the plasma membrane very slowly. And some substances which can easily dissolve in the lipids such as oxygen, carbon dioxide, non-polar organic molecules can easily enter and exit out of the cell. Okay? The plasma membrane of bacteria contain many enzymes, no? So, what's the role of the enzymes? The enzymes catalyze the particular reactions. It catalyzes the breakdown of nutrients. So, what will happen? During the breakdown of nutrients, some energy molecules are released. What are the energy molecules? CITP. So, this energy molecule is released during the breakdown of nutrients. This is another important function of plasma membrane. And plasma membrane also serves as the site for certain metabolic process such as respiration, photosynthesis, then synthesis of lipids and cell wall constituents. So it provides the site for this process. And sometimes this plasma membrane also contains special receptor molecules. So what's the role of these receptor molecules? It helps the bacteria to dictate and respond to the particular chemicals which are present near their surroundings. So this is the important function of the receptor molecules. And plasma membrane is also very essential for the survival of microorganisms. So these are the important functions of plasma membrane. So from these books only I collect the references for this topic. Okay. I think you clearly understand about the fluid mosaic model structure of plasma membrane and the functions of it. So if you find any difficulty in this topic, you can please write your queries in the comment section. Okay? Thank you.
If you like this video, please don't forget to subscribe, share, like and click the bell button. If you have any more doubts, you can write your doubts in the comment section below. Thank you.